Hello and namaste. Welcome to BioMed Diaries. So today we are going to learn about Automatic Kjeldahl Machine, one of the most important laboratory instrument used to measure protein content by determining the amount of nitrogen in a sample. So let's work. If you work in agriculture, food science, veterinary science, dairy, environmental laboratories or biomedical research, this machine is extremely useful. So let's begin. So starting with what is Kjeldahl machine. So the Kjeldahl method was developed by John Kjeldahl in 1883. It is a standard laboratory method used to determine total nitrogen in a sample. Since protein contain nitrogen and the nitrogen value can be converted into the protein content, so the automatic Kjeldahl machine automates this entire process, making the analysis faster, safer and more accurate than
is widely used in
food testing laboratories, dairy instrument, animal feed laboratories, agriculture research center, veterinary and fisheries laboratories, environmental laboratories, universities and research institutes. So the common sample that can be included for the Kjeldahl machine is milk, meat, fish, wheat, rice, animal feed, fertilizer, soil, water and seeds. So the Kjeldahl methods are generally done with mainly three steps. First one is distillation, digestion and titration. So the step first is digestion. So the sample is placed into a digestion tube. Concentrated sulfuric acid act as a catalyst and are added in it. So the sample is heated at high temperature. During digestion, all organic nitrogen is converted into ammonium sulfate. So the distillation starts after digestion. So after digestion, the tube is transferred to automatic Kjeldahl distillation unit. Sodium hydroxide added. This convert ammonium ion into ammonia gas. Steam carries the ammonia into a receiving funnel flask containing boric acid. The ammonia dissolve into the boric solution. So the third step is titration. Finally, the captured ammonia is measured by titration. Modern automatic Kjeldahl machine performs this titration process automatically. The instrument calculates nitrogen percentage, protein percentage. Results are displayed directly on the screen of the display. Why do laboratories prefer automatic system? So this has a high accuracy, fast analysis, reduced chemical exposure, automatic reagent addition, steam distillation, automatic titration, digital result calculator, less human error and better laboratory safety. Although it is a gold standard method, it has some limitations. It measures total nitrogen, not protein directly. Some non-protein nitrogen compounds may affect the result. Strong chemicals like sulfuric acid and sodium hydroxide require careful handling, and digestion takes time compared with some rapid protein analyzer. So in conclusion, the automatic Kjeldahl machine remains one of the most important and trusted laboratory instrument for nitrogen and protein analysis. Whether you work in agriculture, food quality control, dairy product, veterinary or biomedical research, under this instrument is essential. If you find this video helpful, please like, share and subscribe.